Hello, I'm David Lillicrap. I'm a professor in the Department of Pathology and Molecular Medicine at Queen's University in Canada. And I'm going to be presenting to you a slide set that deals with gene therapy in haemophilia. The learning objective of these slides is to describe gene therapy correctly, including basic terms and concepts. This is a story that began now over 65 years ago, and shown on this slide here is a picture of Francis Crick on the right-hand side and James Watson on the left-hand side, pointing at their elegant model of the double helix of DNA that they described in Cambridge in 1953. Fast forwarding now to the early 1980s, when the first of the clotting factor genes were described, and this is a News and Views article published in Nature by my mentor Arthur Bloom, describing the benefits of those cloning experiments with increased understanding of the pathogenesis of bleeding disorders, advancing diagnosis, and ultimately, relating to this slide presentation, genetic therapy. The principle of gene therapy is relatively straightforward. It involves taking a therapeutic transgene, delivering it to cells where the DNA gets incorporated into the nucleus. The nucleus then expresses mRNA, which is translated into protein, and that therapeutic protein either stays within the cell or is secreted from the cell and circulates in the body as a therapeutic benefit. Before I go on to describe the details of the gene therapy process, it's important to raise the issue that currently we're limited to somatic cell gene therapy, that is, either delivering a normal gene or repairing a gene within the cells of an individual and not for the benefit of subsequent generations. So somatic cell gene therapy is limited to an individual, and we are not intending to change or alter germ cell DNA. On this slide here, you see a list of diseases which have always been very good candidates for gene therapy. They're monogenic diseases. That is, the clinical phenotype is due predominantly to single gene defects, and you can see that hemophilia is listed on this slide. Hemophilia is there because we know that small increments in clotting factor protein produce significant clinical benefits. The protein is secreted from the cell, therefore it just needs to get into the circulation. Tight expression of the protein is not required. And we have very good animal models in which we can test these gene therapy strategies in mice and in large animals. So, somatic cell gene therapy. On this slide, you'll see four possible options. The first of them is mutation repair, and I'll describe a little bit more about this on the next slide. Next, we have the possibility of delivering transgenes through non-viral gene transfer. These are still theoretically possible, but aren't close to the clinic. Next, we have cell-based gene therapy, and I'll describe more about this in a subsequent slide. And then ultimately, viral vector gene transfer, which is the strategy that's being used in all currently ongoing human gene therapy studies for hemophilia. This slide then describes the issue of potentially altering genes or repairing genes through gene editing strategies. On the left-hand side of the slide, you see the possibility of homologous recombination as a way of repairing genes. This is theoretically possible, but it's a very inefficient process. And so on the right-hand side of the slide, you see a number of different editing nuclease-based approaches. So reading from the left to the right, zinc finger nucleases, TALENs, or CRISPR-mediated strategies in which protein or nucleic acid-based ways of delivering a nuclease to a region of the genome where a double-strand break is made. Now we're looking at the bottom part of the slide, and that double-strand break is altered or repaired in different ways. These studies are now potentially usable. They're just about to enter clinical studies. But for hemophilia, I think although this can be done in vitro and in animal models, these are some way away from clinical application. So what are the elements of a gene therapy strategy? They're listed on this slide here. I'm going to talk about the therapeutic transgene, a delivery system, an appropriate host cell, and then measurements, outcome metrics for expression of the transgenic protein. So what does the transgene cassette look like? It usually contains a coding DNA sequence or a cDNA sequence which is lacking introns. Most of those cDNAs are somewhere in the range of 2 to 7 kilobases in size. And I'm going to talk about two ways in which you can alter the coding sequence on a subsequent slide. So codon optimization is described on this slide here. It's a change of the nucleotide sequence, that is the triplets of nucleotides that encode amino acids, but there is no change in the amino acid sequence. And the aim of this particular strategy is to enhance the rate of mRNA transcription, to optimize mRNA processing and splicing out of any remaining intronic elements, and finally to match the mRNA translation to the abundance of transfer RNAs that are present in the targeted host cell. So the two other groups of elements that are required in the transgene cassette are some regulatory elements in the 5' end of the transgene. So these are promoter elements which may be regulated on a cell type specific, a developmental specific, or even can be drug inducible. And then more distant elements which are required to enhance gene expression, so-called enhancer elements, which again are usually matched to a particular host cell type. And then at the other end of the transgene cassette is a 3' sequence, which usually is there to stabilize the messenger RNA that is encoded by the cDNA sequence. The next two slides deal with the general strategies for transgene delivery. The first of those describes direct in vivo gene transfer, in which the transgene is delivered often by either intravenous infusion or possibly by a method like intramuscular injection. The advantages of this is that this is practically easy. It's minimal time commitment for the clinician and for the patient. The downsides are that there is in vivo exposure of the vector delivery system to the host immune response. And this can cause some problems, as we'll discuss sometime later. The second thing is that there's variable targeting efficiency. So you may actually be delivering the transgene to some cell types that you prefer not to do. The second strategy for transgene delivery is shown on this slide here. So this is referred to as indirect or ex vivo gene transfer, where the transgene is being delivered outside of the body. So you can see on the left-hand side of this slide, the first thing to do is to harvest or isolate autologous long-lived progenitor or stem cells from the patient. Once those cells have been isolated, you then deliver a normal copy of the gene that is mutant within the patient, or you can edit the mutant gene that's present within the patient's cells. Those genetically modified cells are then expanded outside of the body, and they are then reintroduced back into the patient, where they find a place to reside and produce the normal factor, in the case of hemophilia, the normal clotting factor that's missing in the patient cells. There are a number of advantages and weaknesses of this delivery system. The pros are that there's no in vivo vector exposure. This is immunologically important. It's an advantage. And there's direct cell targeting. So the only cells that see the vector, that the new transgene, are the cells that have been harvested and are accessible outside of the body. The disadvantages are that this is a labor-intensive procedure. It requires special facilities. And also importantly, it requires that the host undergoes a conditioning process to make room for the genetically modified cells to be reimplanted in a successful fashion. So all current hemophilia gene therapy studies involve viral vector gene transfer, and there are three types of viruses that have been used: adenovirus, retroviruses, and adeno-associated virus. I have just one slide discussing adenovirus. There's been one patient treated with this viral vector system. It's highly efficient in transducing, replicating, and non-replicating cells, and you can make large amounts of this vector relatively easily. The significant downsides of adenoviral gene transfer are that there is a significant innate immune response that is mounted to these vectors. So you get elevation of pro-inflammatory cytokines, toxicity of the liver, thrombocytopenia, And in extreme cases, you can develop systemic inflammatory response syndrome, which occasionally can be fatal in nature. So this is not a delivery system for hemophilia patients in its current form. The second viral-based delivery system is that of lentiviral mediated gene transfer, and this is being investigated by a number of groups for subsequent hemophilia clinical translation. Lentiviral vectors transduce both dividing and non-dividing cells. The vector capacity, that is the size of the transgene insert, is pretty reasonable at about 8 kilobases maximum. There are minor innate immune responses, and an advantage of the lentiviral system is that there is stable genomic integration, which appears to be random and non-oncogenic in nature, at least from the studies that have been done, which are extensive in animal models. So finally, the third of the viral vector systems is Adeno-Associated Virus. This is an electron micrograph showing many small AAV particles, and towards the center of the slide you can see one large adenoviral particle. This is the viral vector system being used in all current hemophilia gene therapy studies. AAV, as I'm going to refer to it in the future, is a virus which is a non-pathogenic human parvovirus. It has a single-stranded DNA genome, which is illustrated at the top of this slide. So it's 4.7 kilobases in size. It has two coding regions, a rep gene and a cap gene, and the genome is capped at both ends by inverted terminal repeat sequences. This viral vector system induces minimal innate immunogenicity. There are many different serotypes which are represented by capsid protein variances. A very minor proportion of the recombinant vector integrates into the host genome, and most of the vector exists as stable extrachromosomal circular concatenamers. So this slide basically tells you how you convert a wild-type AAV genome into an AAV therapeutic vector genome. You remove both the rep and the cap genes, and you can see that inserted below in the second diagram that the AAV vector genome now has a therapeutic gene on the right-hand side and a promoter or regulatory sequence which drives expression in a particular organ or cell type. Shown on this slide here, this is a liver-specific promoter which would drive expression of this transgene in hepatocytes. And currently for hemophilia, this is where all of the vectors are being targeted. At the bottom of the slide, you see that the transgene inserts vary between factor IX, where the cDNA sequence is about 1.3 kilobases in size, and for the B domain deleted factor VIII, about 4.7 once the B domain coding sequence has been removed. So how do you make an AAV vector particle? Well, that's described on this slide here. At the top of the slide, you see the transgenic nucleic acid sequence with the ITR sequences, which have been maintained at the 5' and 3' ends of the transgene, the promoter sequence and the transgenic gene in the middle. And then this is inserted into the AAV vector capsid to make an AAV vector particle. And it's these particles that are injected into the patient to deliver the therapeutic material. That's illustrated in more detail here. So you see that the particle interacts with a host cell membrane. For AAV, the interaction with certain forms of receptors is somewhat well worked out, but also there's still some missing gaps in knowledge. The particle is taken up into an endosome within the cell. Eventually, the particle is released from the endosome. The capsid protein is broken down and expressed on the surface of the transduced cell type, and the transgenic nucleic acid is left behind in the nucleus, where it either exists as extrachromosomal concatenamers, or in some instances, and with AAV this is a lesser extent, gets integrated into the host cell DNA. So when you put all this together for hemophilia, the components of a gene transfer strategy involves a therapeutic transgene, which is either a wild type or a modified factor IX cDNA sequence, a B domain deleted factor VIII sequence. You need a delivery strategy, which currently is Adeno-Associated Virus, although there are studies, as I've mentioned, involving lentiviral studies, which are still in earlier development. You need to deliver it to a recipient host cell, and at the current time that's the liver. And then you need to see whether metrics support success of the studies that you've done. And those involve measuring clotting factor levels in the plasma, measuring the annual bleed rates, and the consumption of exogenous factor VIII by patients. And then, of course, you also need to watch for safety considerations. So this slide here represents a version of the Gartner hype cycle for gene therapy. And I'm going to take you from left to right of this slide. So the technology trigger started in the 1970s. And you can see that at the top of the peak here, by 1990 to 1995, we were believing that gene therapy could do remarkable things. And indeed, some forms of inherited immunodeficiency disorder, like ADA deficiency, had been cured by gene therapy. However, by 1999, there was a disaster where a patient being treated with adenoviral gene therapy, Jesse Gelsinger, died from a systemic inflammatory condition. And this indeed was a trough of disillusionment in the development of gene therapy strategies. Nevertheless, over the next decade, from 2000 to 2010, basic science work on vector development and understanding much better about immune response to gene therapy delivery led us to what is currently a plateau of productivity. This really began for hemophilia in about 2010, 2011, with the first prolonged clinical success of hemophilia-based gene therapy and significant biopharma engagement in gene therapy strategies. And so here we are now in 2019 on the verge of a number of phase 3 clinical studies for both hemophilia A and hemophilia B. This is the last slide of the slide set, and it describes the promise of gene therapy for hemophilia. The slide describes at the top the effects of hemostasis with factor replacement, extended half-life factor replacement, non-factor therapy, and on the right of the slide, the possibility of what can happen with gene therapy. At the bottom of the slide, you see levels of clotting factor which can be measured within the circulation. So if you turn your attention to the information at the left of the slide, you see that there are intermittent peaks and troughs of factor levels which correspond to improvements and then reductions in improvements of hemostasis. This is what happens because of the half-lives of the proteins within the circulation. Those benefits were improved with extended half-life factors, EHL factors, and you can see that from the second set of data, the results in hemostasis and the results with the factor levels. And then moving further right to the non-factor therapies, these factors don't improve the clotting factor levels because they're not using conventional clotting factors in the circulation, but they do improve levels of global hemostasis. And you can see that illustrated at the top part of the graph. And then on the right-hand side of the slide, you see what can be achieved with gene therapy. So gene therapy has the promise of elevating the levels of the individual clotting factors, factor IX and factor VIII, to levels within the therapeutically meaningful levels, and those are then persistent because of the continued expression of factor IX or factor VIII from the therapeutic transgenes that have been delivered. And these should be more than adequate to produce long-term maintained prophylaxis with only rare requirements for intermittent clotting factor replacement. It's been my pleasure to present this background to you. I hope that you've enjoyed it, that you've benefited from seeing this information, and thanks for your attention. Bye-bye for now.